Our next speaker is Dr. Rick Huganir from Johns Hopkins.
Well, thank you, and I really want to thank Mike and Catherine for inviting me. I've known Mike since actually before SRF, I think, actually, so long, long time. And I've gotten to know several families here, and several of your families have been to my lab, actually. And so it's really great to see you all again. And I want to say that you changed the way the motivation of the people in my laboratory. So seeing you and seeing how heroic you are has really motivated my Syngap team to work so much harder. So today I'm going to change my title, and this is called Non-canonical splicing of Syngap regulates seizure susceptibility. And this is really sort of a basic science talk, but you'll see why it's important for therapeutics, because we think we have a novel approach to developing a therapeutic. So first I want to just give my disclosures. I was a scientific co-founder and SAB member of Pneumora Therapeutics. I hold equity in Maze Therapeutics, but they do not currently have any Syngap programs, so that there's no conflict there. But I'm founder and SAB member of Syngap Therapeutics, an affiliate of Gondola Bio, and so obviously that's involved in Syngap. And there's several of the Gondola crew here if you want to meet them. The story of Syngap. So this is, now there are dozens and dozens of papers in Syngap, especially these days, but these are sort of the hallmarks, at least in the early phase of Syngap for me. And that is, obviously first identified as a PDZ interacting protein in the brain, and PDZ 95 is a major scaffolding protein in the brain. It's really important in synapse function. And so Syngap, we identified it through its interaction with PSD95 and cloned it in 1998. So that's, how many years ago? A long time ago. And this was also cloned by Mary Kennedy at the same time using a different approach. Also then Morgan Sheng published it's the third most abundant protein in the postsynaptic density. So it's an enzyme, but it's also incredibly abundant, and we think this indicates it plays a structural role at the synapses as well. And then, of course, the knockouts, the homozygote knockouts die very early, postnatally, but the heterozygote exhibit severe synaptic plasticity and learning deficits, which in remarkable ways phenotypically mimic the now known SRID. So, and then what changed the life in my laboratory and has brought all of you together here is that in 2009, the first 3 patients were identified with de novo severe mutations in Syngap and had intellectual disability, autistic repetitive features, et cetera. And now we know this is not a rare disease. We've heard about it. It's become extremely common. And, depend on your estimates, it's up to 1% of neurodevelopmental disease ID cases. So, so one of the first phenotypes, I would just want to give you a couple phenotypes because we'll be using these later in the later slides, looking at therapeutics. That in our initial paper, we can see a plasticity deficit, and this just shows that a well-studied form of plasticity called LTP is deficient in the Syngap mice, the heterozygote mice. So you can see that it has 50% of its wild-type littermate. So that's one deficit. Now, at the time when we were working on these mice, we did not realize they were having seizures or EEG problems. As many of the parents did, when I began meeting with parents years ago, they did not realize their kids were having seizures. And so, but now what we can do is we can look at seizure susceptibility, And it's clear that the heterozygote knockouts are much more sensitive to induction of seizures. And this is a common assay used in epilepsy research. Now on the left I have videos of mice seizing, but I thought I blacked them out so you don't have to watch mice seize. I thought it might not be appropriate for this audience. But what we do is we challenge the mice with a PTZ injection, and this is a GABA receptor antagonist, and it induces seizures within very, very quickly. And as you can see in the Syngap heterozygote mice, this mouse is seizing. You can't see it. But it is seizing, and the wild-type litter is not. And so when we characterize these seizures, you can see, according to, this is a little messed up, but the modified Racine scale for severity of the seizure, you can see the wild-type is basically at a baseline, where the Syngap heterozygote is immediately showing really severe seizures. And then you can look at the doses. If you give dose every 5 minutes after that, lower dose, you can see that over 90% of Syngap mutants seize immediately on the first or second dose, while the wild-type littermate is much more insensitive to seizure susceptibility. So many people in the field use this procedure. And in fact, Gavin Rumbaugh's laboratory showed that this type of seizure susceptibility could be rescued in adult restoration of Syngap protein, giving us all hope that restoring Syngap function later in life can impact these the seizure susceptibility. So today what I want to talk about is the isoforms. And so we heard a little bit about the N-terminal alternative start sites, and we A, B, B, and C, but it's actually much more complicated as we're realizing now. But I want to focus on the C-terminal splicing. We all know that there are 4 isoforms. And in fact, there are more now that we've been getting involved in it. The alpha-1, and this is the isoform that binds PDZ domain of PSD95. It's also the one that we cloned, both Mary Kennedy and my laboratory cloned way back when. But since then, we've others, and we have looked at, found there are 3 other isoforms, alpha-2, beta, and gamma. And so these are due to C-terminal splicing of the C-terminal region. And so this is the structure of the protein, and this is a little complex slide, but here are all the exons with the start sites, all the alternatively spliced exons. And what we want to do is focus in on these C-terminal regions that generate these 4 isoforms. So just if you take the alternative splicing, the beta form is a combination of exon 17 and 18, shown here, and then that produces a stop site. Gamma is 17, 18, and 19, and that produces another stop site, so that's the production of those 2 isoforms. The alpha-2 isoform is 17, 18, and 20, producing a stop site, having different C-terminal structures. And then there are 2, actually, we found alpha-1, which is the one we cloned originally, is just 18 to 20, and it's quite distinct, and I'll talk about this in a second. But we actually found a new novel splice variant called alpha-3. I'm not going to talk about that much today, but it's an interesting protein in itself. So the weird thing about that is when we started studying this, we went to people at Johns Hopkins who study splicing. And we showed them that we wanted to study the splicing, and they said the alpha-1 isoform should not exist. They said, we don't believe it exists. And we said, well, here's the protein proteomics, here's the RNA-seq, and they said, well, it can't exist because it's totally non-canonical. There's no splice donor site in the gene. So just to explain this further, if you look here, so we look at the structure, So normally, if, here we go. So here's the exon 18 going into intron. Normally, you have a GT here, which is the spliced donor. And this is where most 99% of splicing occurs, at a GT donor site to an AG site at the end of the intron. But the alpha-1 actually occurs with one base pair shift. So it's a GGAG. This type of splicing does not occur in any other gene in the genome except for one other protein. And so it's incredibly unique, a very unique splicing event. Interestingly, this leads to the C-terminal PDZ domain. So this is really critical for binding to set PSD95. Just for your information, the alpha-3 also is a non-canonical splice product, but I'm not going to be talking about that as well. So when you look at the distribution of these, the alpha-1 is really 30% of the Syngap in the brain. So this is a large amount of this splicing that's occurring in brain. It's actually specific for brain. It does not occur in other tissues. So this just shows that it doesn't occur in heart, liver, testes. Mostly these are the beta and gamma isoforms. And this is conserved both in humans and in mice and many other species as well. Now, so this led us to believe that we should look at the function of these, and many people had indicated that the different isoforms had different functions. And so back in 2020, we knocked down Syngap in cultures, and we did this chemical LTP protocol that was discussed earlier. This is sort of looking at LTP, the amount of receptors at synapses before and after induction of chemical LTP. This is your control. If we knock down Syngap, you can see you lose this regulation by chemical LTP. If you rescue with alpha-1, you rescue pretty well the LTP as shown here. But it turned out the other isoforms did not. Alpha-2 maybe barely, but not really as well as alpha-1. And this is consistent that alpha-1 is really critical for plasticity and agrees with work shown on the right from Gavin Rumbaugh's laboratory, where he made a knock-in mouse which mutated the PDZ domain, just put a mutation in there so it does not bind to PSD95. So this mouse is totally normal, it has normal level of alpha-1, but it cannot bind to PSD95. And he finds that this mice has the same phenotype as the heterozygote knockout mice as shown here. They're looking at LTP. So this led us to have a therapeutic hypothesis that maybe if we could regulate Syngap splicing to increase alpha-1 Syngap isoform, this might be a therapeutic approach to treating kids. Syngapians. I've been around long enough I can call you Syngapians. But first we got to understand how this non-canonical splicing works. And so we started investigating this, and this is the exon 18. This is where all the splicing occurs over here. And remarkably, this is an intron, and introns are not normally conserved. But within this region of Syngap, it's totally conserved against many, many different species. And this region you'll see is really important for the splicing, this non-canonical splicing. Now, the only species that does not have it is platypus. So we don't really understand this. We haven't gotten any platypuses in the lab, but the platypus does not have this conserved region and presumably does not have alpha-1. So a great talented student in my laboratory, Tracy, has worked with Inge and Rich Johnson. Many of you know Inge Hong. He's really been a leader in this in my lab in this area. Many of you have interacted with him and known him. Sadly, he's gone back to Korea, so he's no longer in the lab, although we continue our collaborations. So they developed a splice reporting system where we could actually analyze the mechanism of the splicing. And I won't go into detail, but this is a system where you can transfect this into neurons because the splicing only occurs in neurons. And if it turns, the neuron turns green, it's getting alpha-1 splicing. If it turns red and mature, it's going to alpha-2 and alpha-3 splicing. And if it goes to gamma, it's just it's blue. And so I won't go into the details of that. You can ask me later. And then we did sort of classical structure function analysis of the intron. And so we took the intron between exon 18 and exon 20. We did deletion analysis, and we found that deleting this small region here eliminated alpha-1 splicing. We further deleted regions and made smaller deletions, deleted the regions of that first part of the exon and the intron. And you can see we get, again, a dramatic decrease in alpha-1 splicing. We then went on to actually do a further deletion of just a very small 26 base pair region and eliminated alpha-1 splicing. So this region is really critical for alpha-1 splicing, and it narrowed us in to do further studies. So we went on to do point mutations. So here we're doing single point mutations within those regions. And this, making single point mutations, we could dramatically decrease alpha-1 and several in the same region. So several different mutations shown here. And you'll see that later this is really important for our studies down the road. Now, the question is, can we develop a therapeutic? And so the obvious thing is, can we develop an ASO which will upregulate alpha-1 and therefore possibly increase alpha-1 expression, and then will this have a therapeutic effect? I'm not going to go through antisense oligos. I think most of you are familiar with this. But this would be called a splice switching ASO. And we did the classical ASO walk. So we created a variety of ASOs that flanked this region that we thought was very important. And what we found is actually several ASOs that upregulate, in this case, 2 to 3-fold the amount of alpha-1 splicing using this reporter system. And it turns out it borrows from alpha-2, so it switches takes away from alpha-2 and increases splicing to alpha-1. This also is shown here. So dose dependent. On the left, it's looking at the normalized EGF signal. On the right is amplicon sequencing, showing again dose dependent effect on alpha-1 splicing using the system. So we can regulate isoform splicing in the system. We've gone on now, and I don't have time, but we've actually been starting to do in vivo studies, injecting with our lead compounds, and we're seeing increases. Preliminary data is really showing increases in both RNA and in protein expression in vivo in the intact mouse. Now, does this splicing really occur in vivo? And is this region that we characterize really occur in vivo? And so we took one of those mutations, those point mutations, which eliminated alpha-1 splicing in that system in cultures, and made a mouse. We knocked in this mutation in the mouse. And so here we have a mouse that has basically the homozygote knock-in has no alpha-1. It's all alpha-2. So we would predict that this mouse should be show some of the phenotypes that the Syngap heterozygote does. There's no effect on other splicing of beta, for example, but it turns out it does impact seizure susceptibility. So here, if you look at the wild type is the black, you see that again it takes The first dose, you don't see much response. The heterozygote knockout, which only has half of alpha-1 expression, there's a slight shift. You'll see it's more dramatic in the next slide. But a homozygote, so this is a mouse that has normal levels of Syngap, but no alpha-1. So it's seizing immediately. So, and here's the dose dependency. Again, so the wild type takes many. The heterozygote takes less doses to seize. And this mouse that has no alpha-1 seizes immediately. So we think alpha-1 is special, not only special in the way it's splicing, but it's also special for rescue of the mouse. And so we are, charging ahead, trying to develop this as a therapeutic. This is a very novel approach. You're regulating splicing, not levels of total protein. And so it's going to be relatively safe. We won't be overexpressing protein. So alpha-1 is expressed in brain and conserved in many mammalian species. It's the first 26 amino acids, base pairs of intron 1 is essential. The splice reporter shows efficient interrogation of these requirements. And then ASO walks reveal lead ASOs that we're developing with Therapeutics and in collaboration with NLORM to really get very effective ASOs to do the in vivo studies. And alpha-1 isoform displays seizure susceptibility. Now, we've also been taking another approach, and I want to just mention this because it confirms our hypothesis that alpha-1 is really critical for this rescue. And you'll hear much more about this in the next talk from Megan. But what we did is we packaged all 4 isoforms into AAV, and we can get very good expression of all 4 isoforms. It's shown, this is alpha-1. And it's very highly expressed at synapses. And so we inject it into mice. This is the overexpression of alpha-1. And we can, this just shows all the different isoforms we can express. But then we started doing assays, and we looked at activity in the open field, as Ben just mentioned, is one of the assays that we use. And we can totally rescue this increased activity in the heterozygote knockout with a rescue with the AAV. And we also see changes in EEG, rescue of the EEG properties as well. So in closing, this is the Syngap team. Ingi made this slide, and I think he put every undergraduate who walked into the laboratory while he was doing an experiment on this slide. But the key people have been Ingi, Colin, Richard, Tracy, and Yoichi. And many of these other people have been involved in the laboratory, but many of them have been just passing through the labs. So funding, this has been the funding in the past. I left Syngap. Mike said I could say Syngap Research Fund because I've been here before. It was SRF, but I will change this next time, Mike. So this is our funding, although it's getting rather short these days, as many of you know. So I'd be happy to take questions.